All right, I'm going to take you through how to hopefully fairly efficiently read an article and understand it. So I'm going to go through this fairly quickly, but I think this is an extremely important thing to know how to do. One, you're going to be reading articles in this course for presentation as part of your units. And two, you're going to be reading articles throughout your whole life and scientific career. So it's really important to kind of have an idea of how to do this efficiently. Now, that doesn't mean you're going to be able to read through these articles very, very rapidly. It still takes me a very long time to read a primary literature article. But if you kind of have an idea of the way you should attack that article, the way you should go at understanding it, I think it can be made a little bit more efficient. And then in the second video, I'll give you an example of kind of how to present so that when you're presenting your article for this semester, you'll have a little bit of the ideas of do's and don'ts that you've probably heard along the way, but I think it can still be a good reminder. All right, so when you get to the article, the very first thing is just try and figure out what it's doing and what its purpose was. And you really can get that through the title and the abstract. So again, the first thing we're going to figure out is what the main question, what the main purpose of this article was. What's the question they were trying to answer? Within that, you'll kind of have the hypotheses, and also in the abstract, you can get the overall conclusion. And this is going to be a very important theme that I'm going to go over. If you have an idea of what the authors were trying to do, and then what they think they've accomplished, what do they think their data showed, then understanding the figures and the data itself becomes a lot easier, actually, because you already know what they think they've shown, and those figures are going to show those parts of those conclusions. So you really want to have the end take-home message in mind when you're reading through the rest of the article. And while you're in that abstract is really a key overview of the whole article. So even though the abstract is one of the shortest parts, it's worth spending a little bit of time to understand what the abstract's telling you, because that's going to give you an overview of the whole article. And so if there's some terms in the abstract that you don't understand, it's worth looking them up now, because those terms, if they're important enough to be in the abstract, they're going to be seen over and over again throughout the article, and chances are good they're very important to understand what those terms are, what they relate to, and how they relate to the article's purpose if you're going to understand the article itself. All right? And then obviously, if you're assigned this article, you don't have much choice, but from the abstract normally you can decide if it's worth your time to read further. All right, so let's take a look at this abstract together. So this is the COVID article that you can download. All right, and I've just got it up here in a PDF format. I'm going to have you pause the video every once in a while to really do some of these things. And so I first want you to read through the summary. Basically, summary is synonymous with abstract. That's the term Cell articles use. And what I want you to do is open up your activity document, and you're going to figure out the purpose of this. That's the main thing. And you're going to figure out the conclusions. And I want you to look up a few key terms that you may or may not understand. All right, so go ahead and do that through the activity sheet for a second. All right, so hopefully after kind of reading through this and maybe doing a little bit of extra research, and simply by extra research, I often mean just go to Wikipedia or things like that. But hopefully by now you probably know SARS-CoV-2. That's an important term here, but you probably understand that already. That's the virus that causes COVID-19. But you may not have heard of ACE2. You may not have heard of TIMP-RS2. And so I just simply looked those up. And with TIMP-RS2, I found that this is a serine protease member. So protease means it cleaves proteins. So that's probably going to be something important. And it is transmembrane. And ACE2, when I look that up, a lot comes up because it's been determined that this is the membrane protein on the cell that the coronavirus binds to. So it's really a popular topic of research right now. But even without that, I could kind of figure out by Wikipedia, it's an enzyme attached to the cell membranes. Oh, this might be important. It's in the lungs, arteries, heart, kidney, and intestines. Those seem to be the tissues that COVID-19 affects the most. All right, then it has a normal function. Obviously, ACE2, we know it's the receptor for COVID-19, but it's not put on the cells so that COVID-19 can get in. It had a normal function as well. And so that might be important to understand. And so you can start to get a feel for the overall importance of some of these different factors here. And really it says in the end, let me highlight here, there's a couple of things that are important. We show that the sera from convalescent SARS patients cross-neutralized SAR2-driven entry. All right. Also, before that, they demonstrate that the SARS-CoV-2 uses ACE2 for entry and TIMP for priming. So what this means is that they have figured out that ACE2 is the receptor, TIMP-RSS2 is also important for the viral entry into the cell. And from this, we can pretty much figure out what they are going to do. They are basically trying to figure out how COVID-19 enters the cell. That's the question. How does COVID-19 enter the cell? They figure out that its receptor is ACE2. It needs to be primed by this TIMP-RSS2. And then their other objective was to see if they can figure out ways to block it. And one of the conclusions they had there is that sera, in other words, serum from patients that have had previous SARS, might be able to help patients with this new COVID-19. And so all these things we can get from the abstract, and that right away is going to give us a little bit of insight into what this whole article is going to be about. And we'll already start to make understanding those figures easier, because those figures should point towards the fact that ACE2 is the receptor and TIMP is needed for priming. So whatever figure results there are, they're going to support that information, and then probably towards the end you'll have some figures that test whether or not sera from patients that had other types of SARS virus can actually prevent SARS infection into cells. I already know what to expect from the methods and from the results, and now understanding each figure with that idea in mind, that goal in mind, if you will, is going to be a lot easier later on. All right, so let's jump back over here. So we've got the idea. Now we can go to the introduction, and that makes sense. It's the next part there, so you naturally would read that right away. But this is going to give you all that background information. So what was already known? What background information did they use to get to this idea? So chances are good they didn't jump right on ACE2 as the receptor. Chances are good there was something previously that gave them this idea. So what was that? All right, and it'll also give us a little more context. All right, so why did the authors perform this work? What was their main objective? What's the relevance? Now, that relevance is pretty easy to see right now with COVID-19. That relevance is the fact that hundreds of thousands of people, unfortunately, around the world have died, and this is disrupting our lives, so there's huge relevance. But even other research that isn't as currently prominent as COVID-19 is going to have a reason for doing that. What's its purpose? Why did the researchers think this is important? So what was the background, and why was it important? All right, so let's jump ahead back to the article here for a second. All right, so continue to follow along and do the reading articles efficiently activity. And so at this point, I want you to go ahead and read the introduction portion. All right, so it just kind of starts here, goes through the second column, jump over figure 1 for now, and then kind of ends at this point here. So within that, you're going to help put this into context. All right, so why are they doing this? And I want you to also see if you can find the reasons the authors were focused on ACE2 and TIMP-RSS2. That is all in the background here. See what you come up with. Again, pause the video. All right, so let's take a look at this together. Hopefully you've had a chance to read through it before you're continuing on. If not, again, pause the video and do that. But this first part here is really talking all about why SARS-CoV-19 or SARS-CoV-2 is important. And I think we all have a pretty good idea of that currently, but it does give a little bit of background. If you somehow had been living under a rock and you didn't really know where COVID came from and things like that, it gives you that background along the way. But the real important part that you probably don't necessarily know starts here. And so in this portion here, they're really talking about previous SARS viruses. So this is not the first SARS virus we've seen. In fact, that's why it's called SARS-CoV-2, because there was a SARS-CoV-1. So the original SARS infected a lot of people many years ago, and then MERS as well are both part of that coronavirus. And so they're looking for the similarities and differences between these viruses to see if we can learn from some of the knowledge that we have in the previous viruses to really attack this COVID-19 virus. And it says here that in those, right? Those earlier viruses, they knew that you needed to bind, the virus needed to bind, and it used ACE2 as the entry receptor, but it also had to cleave that viral protein. It had to cleave the spike protein. So the spike protein on the virus is what it lets it bind to ACE2, but that had to be cleaved in order to allow fusion of the virus. And this is kind of something that I talked a little bit about in the first video. All right. And so since the previous work on the previous SARS viruses identified these as the receptor and a key protein, TIMP-RSS2 that is, for fusion, they're going to see if it's the same here. All right, this next step might be what's new for some of you. If you're like me when I was in undergrad, I basically just read a primary literature article from start to finish, and by that time I was overwhelmed, extremely confused, and I really wish someone had told me something like this then. Others of you might be smarter than me and already do this, but next I suggest jumping from the introduction to the discussion. Skip right past the results, right past the methods section for now, jump right to the discussion section. And there's a few reasons for this. One of those reasons is that here we're going to find the full set of conclusions. Almost always within the first paragraph or two of the discussion is going to be a list of what the authors think they've said. Right? So the whole article format kind of starts broad. Right? The introduction starts broad and then ends with exactly what they're doing and why. And then the discussion is going to be like the other side of our hourglass. It's starting with exactly what they showed, and then they'll put it back into context. So we want to figure out exactly what they think they've shown. We have a broad idea of it just from the abstract in the introduction, but this is often going to be a little bit more specific of step by step what they showed along the way and how they reached the overall conclusions. We're also, in the rest of the discussion, going to see if they found any surprises or were their hypotheses correct. We'll also learn a little bit about what further work is required. This is actually important for y'all. Science is never done, so what things are important to address next? What things did they not yet address in this article? All right. And then finally, it'll put it back into context. But what we're really interested in order to understand this article is what the main conclusions were and what's still needed. So here we are back at the article, and I'm going to jump from our introduction straight to the discussion section. Right? And so again, following along and working on this activity, I want you to go ahead and do the discussion portion on your reading articles efficiently activity. And I want you to see if you can find those 1 to 3 sentences that really say what they think they've shown. What are their exact conclusions? What do they believe that their data conclusively allows them to state in terms of the conclusions of the article? All right. Then I want you to see if you can find a few unanswered questions. What are some of those next steps that would be? And then this part's relatively easy for this article, but what do they think they've achieved? What do they think the biggest aspect of their results are? What are they, how are they contributing to the overall field? What's the importance of what they've found? All right, so go ahead and read through the discussion and see if you can find those things. Their main conclusions, next steps, and relevance. All right, so let's talk about the discussion a little bit together. So right off the bat, you pretty much get their main conclusions in the first 2 sentences. The present study provides evidence that host cell entry of SARS-CoV-2 depends on the SARS-CoV receptor ACE2 and can be blocked by a clinically proven inhibitor of the cellular serine protease TIMP-RSS2. So CoV-2 uses the same receptor as the original SARS virus from a couple decades ago, ACE2. Moreover, it suggests that the antibody responses raised against SARS-CoV could at least partially protect against SARS-CoV-2. So because they use the same receptor, people that have antibodies against some of this original SARS, they might actually be able to help people defend against this new SARS if they can block that virus receptor binding. And then they talk a little bit about the TIMP-RSS2. And so they know that that's important because if they block it, the virus doesn't get in. So these are key things that we are going to look for in the results. We're going to look for the similarities of the spike protein that binds to ACE2. There must be similarities from the original CoV and CoV-2 in order for them to be able to both bind the same receptor. So chances are very good they're going to show similarities between the spike proteins of the two viruses. Chances are also good they're going to show that the virus needs ACE2, needs the receptor to get in. And chances are good, and actually they state it directly, that they're going to show that it also requires priming. If you don't have TIMP-RSS2, then you reduce viral entry. And then finally, chances are good that they're going to show that they took blood from patients that had immunity against the original SARS, and sometimes that blood could at least reduce the infection because, presumably because, it was blocking the interaction of the virus with ACE2, since the two old coronavirus and new coronavirus both at least use the same receptor. All right, so what about some of the other things? Well, the next steps. These are a little bit harder, but there's often words in there that help. I've underlined it here. So one may speculate that the new virus might exploit cellular attachment promoting factors with higher efficiency than SARS-CoV-2 to ensure robust infection of ACE2. That's a mouthful. I realize that's a mouthful, but when you see one might speculate or it remains to be seen or anything like that, that's indicating next steps. So yes, this virus uses the same receptor as the original, but why is it so much more powerful? Why are we having such a problem with this one that we didn't with the other one? So the original SARS virus was actually very deadly, but it didn't infect very fast. And so basically it only infected 1,000 people or so, and then it was gone, as obviously opposed to what we have here. So why is that? Why, if they use the same receptors, does COVID-19 transmit so rapidly? Well, that's a next step. They need to figure that out. And they have speculation, they have hypotheses, right? Maybe there are, there's more efficient binding or higher affinity binding with this virus, but they still need to find out. And there's a few other places they talk about it. All right, so here's another one can speculate. One can speculate furin-mediated precleavage helps. All right, I don't want to spend too much time on this. That becomes a little bit more understandable if you kind of keep reading the article. But there's probably more than one way for it to get in, because they show that TIMP-RSS2 helps, but even without it, there's other ways. So they need to find the other ways. And we still have another one can speculate along the way. It's all over the place. Here we go. Although confirmation with infectious virus is pending, our results indicate neutralizing antibody raised against the original CoV could offer some protection against CoV-2. Now, they've shown that in cells, but they need to show that in clinic. So a next step is seeing if that actually works in clinic. A lot of these next steps, because COVID-19 research is happening so fast, have actually been answered. And if you're interested, I'll have a kind of summary video that summarizes a little bit more what we know about COVID-19. Why is it more infectious, and can it use some of these other ways for priming and infecting? So if you're interested, you can watch that video. That one's optional. But the take-home message here on the discussion is that we have direct statements of what they've shown, their conclusions. And I didn't talk about it earlier, but they even go into exact data, which are in the results. Right? SARS-2S was efficiently cleaved in cells, and cleaved S protein was incorporated into VSV particles. Right? We'll talk more about VSV particles. But this is an exact result from one of the figures. Right? And then down here, the present study indicates that SARS-CoV-2 also depends on TIMP-RSS2 activity. We note, however, that SARS-CoV-2 infection was inhibited but not completely gone by that TIMP-RSS2 inhibitor. And this is why they think there must be another entryway. But this is an exact result. So somewhere in the results, they must show that TIMP-RSS2 blocks entry, but not completely, meaning there's something else. So again, these conclusions are really important to pull out of the discussion so that you can better understand the purpose of each figure and what the results of each figure, the take-home message of each figure along the way. And then those things like it thus remains to be seen, or one can speculate, or it's yet unknown. All those things are telling you those next steps right off the bat. All right, so at this point, I'm going to go ahead and stop this video. Hopefully you've been able to get through most of the activity as we go through together. And then I'm going to come back in the next video and I'm going to go through a couple of the results. I'm not going to go in great detail through all of them, but I'm going to go through how you can use this information that we've garnered from the abstract intro and discussion to understand the results, and then how to present a result figure. All right, so watch that next video when you're ready.